Okay, so so when you're setting up the test cross for the for this here, you really have, it's a 2 by 1 Punnett square, where this parent, the only combination is B plus, VG plus, or the B and the VG. This parent either passes this chromosome or this chromosome, but either way, it's a B and a VG. So therefore, we have two different kinds of kids, one that's heterozygous for each trait, and one that's all homozygous recessive here, like this. So we have a 1-to-1 or 2-to-2 or ratio here. So 1, this is going to appear gray and normal wings. To this one would appear black and vestigial wings. So that's your phenotype ratio that we should get. And so those are the only two types of kids. And notice that these kids look like one or two, one of their parents. So literally you would expect 50% of their kids to look like this one, be gray and normal, and the other half look like this parent and be black and vestigial. So what Morgan did is when he made it his fruit flies, looking at this trait, gray versus black bodies and normal wings versus vestigial wings, and he made it as fruit flies. He didn't get the ratio that you expect with unlinked or linked, because in unlinked, you see what it's a 1-to-1 to 1-to-1 ratio, and you see all of the different phenotypes, which he did see. This was his, on the bottom of the page, it shows you his actual data. So this is his actual data. He did see 4 different kinds of phenotypes, but it wasn't a 1-to-1 to 1-to-1 ratio like you'd expect if the genes were unlinked. Then, when looking at if the genes are linked, what ratio would you expect? You'd expect a 1-to-1 ratio and see gray and normal and black and vestigial, but yet he saw 2 different ones, the gray and vestigial and black and normal. So therefore it didn't quite fit into unlinked or linked. Can somebody tell me what must have happened to cause his data here? You got way more of these than these, but he saw all 4 of them. So how can we explain that?
Crossing over.
Crossing over. So in what parent would crossing over had to have happened? This parent or this parent? The heterozygous parent. And so let me go over and just explain that and review that with everybody. And so when you're looking at this parent here, let me do 2 different colors here. Let's just look here. So looking at this parent here, when this chromosome replicates to go through meiosis, so here's your replicated chromosome. So it has the B plus gene, and this side is an exact replication, and then the VG plus gene and VG plus. And then when this chromosome replicates, the BVG chromosome, it replicates, and I'm going to draw the arm here kind of long just to show the crossing over here. So they cross over, so this has B and B, VG, and here's the VG gene here. So remember when crossing over occurs that they literally, wherever the point of crossing over occurs, from that point to the end of the chromosome, they exchange DNA, and therefore any genes that are on it. So during meiosis, when these chromosomes separate and are pulled to different sides of the cell and eventually end up in different cells, this green chromosome looks like this with a blue tip here, and that blue tip is going to have the VG gene, and none of the rest of the chromosome is affected. So this is going to be B plus and VG plus. And then your blue chromosome will look like this, where it has the green tip with the VG plus gene, and then the rest of it's not affected, so it would have the B and the VG genes. This is what they look like. And so then during meiosis, the homologous chromosomes will be pulled to opposite sides of the cell, and they'd end up in 2 different cells. So these will be the cells, the two cells at the end of meiosis 1. Meiosis 2, the sister chromatids are pulled apart. So this chromatid goes to one cell, this one goes to another cell. So I'm just going to draw a line here to represent the splitting of this, and this will be in this cell, this combination will be in this cell. Draw a line here, pretend like it's splitting into two. This will be in one cell, this will be in another cell. So these are 1, 2, 3, your 4 cells at the end. Notice that these two are new combinations that weren't possible before. Before you could only have these two combinations, but because crossing over, you have new combinations. So really, then with crossing over, if crossing over does occur, your box would really add 2 more like the unlinked genes. All right, so there'd be 4 possible combinations here. But then why isn't it a 1 to 1 to 1 to 1 ratio like unlinked genes? So we see all 4 combinations due to crossing over, but these 2 are way less than these 2. Why would that be? Why wouldn't they be a 1 to 1 to 1 to 1 ratio? Do you have an idea? Notice what's true about these two. The ones that they get the majority of them, these are the ones that you'd expect anyway, right? And these two are these two only can happen if crossing over did occur and you get these 2 new recombinations. Roman? Okay, yeah, and that might be a better hypothesis if you didn't have a lot of numbers. He's got like a couple thousand flies here, like he's got a lot of data, and so just due to it randomly doing it like that, it wouldn't. While that's why these 2 are not exactly the case. So it's a good hypothesis, but with so much data, that's probably not just due to the random independent assortment. What's to do with crossing over? Does crossing over have to happen every time between those 2 different genes? Absolutely not. And so therefore, sometimes crossing over will occur between the 2 genes and the genes flip and you get a new combination. But a lot of times it might not. And so therefore you get a lot of what you'd expect with not crossing over, because crossing over doesn't happen every time. And so that's what explains that. And so Morgan, so he added a lot to this genetic puzzle. He was the first one to assign a gene to a particular chromosome, and he figured out that some genes are on sex chromosomes and the whole sex-linked trait. And then he said, okay, well, there's multiple genes on the same chromosome, so he's the one that came up with linked genes and that they must be inherited together. But based on his data with fruit flies, he's the one that said that the chromosomes must not always stay intact, that they must switch some spots. So he's the first one to actually come up with this whole crossing over thing to explain his data here. All right, and so that's what Morgan did. All right, and so what we can do, we'll see, is that we can use this data from genetic experiments to do a couple things with it. So we're going to learn a couple things here today. All right, so I'm going to have you go back to your notes. So we're going to finish 15.2. Alright, so we did this first together. We defined what linked genes are. So this next little part of the section just kind of goes over what I just went over with you. So there's some pictures and things like that of Morgan's crosses with gray and black body and normal and vestigial wings. I just had you do it on your own to figure it out on your own because I thought you'd understand it better, and then just looking at the pictures. So this little bit here is just a review of and showing you some pictures of what we just did. So Morgan did these experiments with fruit flies to see how linkage affects inheritance of two characters like body color and wing length. So he crossed the flies that differed in traits of body color and wing size. So this picture here is what you guys did on that sheet on your own. So here was the first cross you did. Gray, normal with black vestigial. You got their kids that are all heterozygous. You did a test cross with them. When you got that, you get these 4 phenotypes. If they're unlinked, you'd expect a 1-to-1-to-1 ratio. Linked, these guys ended up to be linked, and so we still see all 4 phenotypes, but we get mostly these 2 that look like the parents, and just some of these due to recombination. So looking down here, it says these guys are parental type offspring. So these 2 types here are called parental Y because they look like either one of the parents. Okay, so that's parental offspring. And then these 2 right here, they're called recombinant offspring because the DNA is recombined. And so these 2 were only could only happen. These guys here are due to crossing over. So without crossing over, we wouldn't be able to get those 2 types. So that's recombinant. And so usually when you get data like this, where overwhelmingly you have 2 of this kind and then you have less recombinants, that usually means that they're linked genes and that crossing over has occurred. We'll see that here. So from the results, Morgan reasoned that body color and wing size are inherited together in specific combinations. That's the parental phenotype. So they're on the same chromosome, they're going together to the kids. However, non-parental phenotypes were also produced, and those were the recombinant phenotypes. So understanding this led him to explore genetic recombination and looking at producing offspring with different traits than either one of the parents, and looking at the reasons for that, which, as we talked about, is crossing over. So if no crossing over occurs, this picture is just showing you what we already talked about, that this is parent number 1 that's heterozygous, parent number 2 homozygous recessive. So basically, with no crossing over, you'd only expect 2 kinds of kids. This parent's always going to give a B and a VG gene. This parent could give the B plus VG plus chromosome or the B VG chromosome. And so therefore you'd expect only 2 kinds of kids, but notice that both of the kinds of kids look like the parents. But he saw those recombinant offspring. So the genetic findings of Morgan as well as Mendel have to do with the recombination of chromosomes. So unlinked genes, that's what Mendel did. So when we did all the crosses with Mendel, the round and wrinkled and yellow and green and purple and white flowers, those were unlinked genes. And with Mendel's experiments, he saw some offspring that differ from either parent. And then he saw some with the phenotype matching some of the parents. So the parental phenotypes are called parental types, the ones that match the parents. The ones that are different than the parents are called recombinant types, or sometimes shorthand are called recombinants. And so you can see this with linked and unlinked traits, which is what we saw in the thing that we just looked at. And then a 50% frequency of recombination is observed for any 2 genes on different chromosomes. We're going to talk about this frequency of recombination here in a second. And so when Mendel crossed yellow and green and round and wrinkled, and we did a dihybrid cross, he saw a 1-to-1-to-1-to-1 ratio because his genes were on different chromosomes. But he did see parental types and recombinant types, but they were just a 1-to-1-to-1-to-1 ratio. Whereas Morgan saw all 4, but the parental types were way more in abundance than the recombinant types, which then led to the linked genes with crossing over. So Morgan discovered that genes can be linked but the linkage was incomplete, as evidenced from those recombinant phenotypes, the ones that don't look like the kids or parents. Morgan proposed that some process must sometimes break the physical connection between the genes on the same chromosome. What's that process? Crossing over, right? That mechanism was the crossing over of homologous chromosomes and the switching of the DNA, right? And that's what this picture shows you here that I just drew out and went through with you together. So so when you look at this picture later, so you can relate it to what we did, let me just show you what it's showing you here. Here's the test cross. So here's your heterozygous parent with the homozygous recessive. They're showing you the DNA replicating in meiosis and showing you what happens as a result of crossing over. So they switch the VG plus and the VG genes. And then at the end of meiosis, you get 4 different kinds of gametes. These two are the recombinant ones that are only due to crossing over. If crossing over hadn't occurred, you'd only make these 2, okay? This one here is the homozygous recessive. Could crossing over occur between these 2 chromosomes? Yes, they can. Any homologous pair of chromosomes crossing over can occur. If crossing over occurred between the B and VG genes here, would it make a difference though? No. Hence, they didn't show it. Okay, but I just wanted to point out they could. So then, this is your Punnett square and the possible kids, and these are his actual data, so you get way more like the parental than you get the recombinant. Let me introduce you to something called the recombination frequency. Recombination is crossing over recombined genes. Frequency, how often does this occur? So what he did is we add up the total number of offspring. So when he did this cross, he made it a lot of flies. He got a lot of data here. He got over 2,000 flies. So he's counting and tallying up what traits they have and so on. So we take 965 plus 944 plus 206 plus 185. That gives us a total of 2,300 total offspring. So you get 2,300 from this cross. Lots of data. Then what we want to do is said, okay, how many of those are due to crossing over? How many of those that wouldn't occur without crossing over? And that's these 2 boxes here. And so we took 206 plus 185. That gives us 391. So 391 recombinants divided by 2,300 total offspring. You get a decimal, you multiply it by 100, and you get a percentage. So about 17% of all of his offspring that he got from this cross, 17% were recombinants. How did they get to be recombinants? Because of crossing over. So therefore, crossing over in this cross must have occurred about 17% of the time, all right, to get those. And that's called the recombination frequency, or it just basically tells you how often does crossing over occur between these 2 different genes. And so that's going to be important here in just a second. So this, from that, we can make, if we're knowing recombination frequencies, we can make a map called a linkage map using this recombination data. So Morgan's student, Sturtevant, used the recombination frequencies. He constructed a genetic map, which was a list of the genetic loci, which is location, along a particular chromosome. So he could map out where genes are at in relationship to one another along a chromosome. And he predicted that the farther apart 2 genes are, the higher probability that a crossover would occur between them, and therefore the higher recombination frequency. Let me draw what that means here. So if I have a pair of chromosomes, replicated chromosome here, so here's a pair, and let's say I put the B plus gene right here on here, and then I'll put at the end here the VG plus gene. And this one here has the VG gene and the B gene, and the B gene's up here, and the VG gene. So these are your 2 chromosomes. For crossing over to occur and to get a new combination, what needs to occur is that these 2 arms have to cross over somewhere in between the VG plus gene and the B plus gene. So the spots, the the room in between the B genes and the VG genes, these two arms can cross over at any point there, so they could cross over here, 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 here. Because remember, wherever they cross over, from that point on to the end of the chromosome, they're going to switch DNA, right? And so therefore, if it occurs just above the VG gene, and they cross over, they're going to switch the VG genes, and you're going to get a new combination because it's going to be B plus and VG. Or it can occur here, and it's still going to switch the VG genes here, and so on. There's lots of space. Look at how that's different if I draw the two chromosomes where the B and the VG genes are close together. So let's say here I make the B plus and the VG plus here. Okay, so this one's B plus and VG plus. So they're close together on the chromosome. And then this is going to be B and VG and B and VG. So they're real close together. Now, in order to switch the VG genes, what needs to happen? How do I get a new combination? Crossing over has to occur between the genes, right? So therefore, when these, just right through here, so when these two arms cross, where they cross has to be in just that little area here. So the likelihood it's going to be in that little area goes way down, all right? Here, when these arms cross over, remember it's random, since you have way more space for it to cross over and way more, then it's going to occur more frequently, all right? Here, not very frequent because the space of the spot where they actually can cross over and switch the genes is so small, so limited, that the amount of crossing over and the likelihood that those two arms are going to cross over in that little spot goes down dramatically. So that's what this bullet is saying here. It says that if crossing over between two genes you find out happens a lot, that probably means that the genes are kind of far apart on the chromosome. If crossing over, if only occurs, you find out that it only occurs, like 1% of the time, it doesn't occur very often, that probably tells you the genes are pretty close together, because so the frequency is really low. And so that's what the Morgan student came up with. So then he was able to, from that, make a linkage map, a genetic map of a chromosome based upon recombination frequencies. So distance between genes can be expressed as what we call map units. One map unit, or centimorgan it's called, represents a 1% recombination frequency. So what this is, so really it's a frequency. So if recombination occurs between two different genes 17% of the time, that would be 17 map units or 17 centimorgans. It all means the same thing. And so what this does is it indicates relative distance in order of genes on the chromosome, not the precise locations of genes on the chromosome. And so it basically says, okay, and crossing over occurs a lot between these two genes are farther apart, all right, and if it doesn't occur a lot, then they're closer together. So it gives you relative distance, but it doesn't tell you exactly where those genes are on the chromosome. So let me give you an example here. This is a linkage map, but how the Sturtevant made this linkage map is he studied crossing over and looked at different genes. We added the third gene here, the Cinnabon gene here, so Cinnabar, I should say, B, CN, and VG. What he was given is in his experiment. So this is the 17% is the one that we just figured out with the body color and the vestigial wings. That recombination occurs 17% of the time. Between the B and the CN gene, recombination occurred 9% of the time, and between CN and VG, recombination occurred 9% of the time. This is the data he was given, and from this data, he made a linkage map based upon the fact that the higher the recombination frequency, the farther apart the genes are. So he took the farthest apart here, so B and VG. So B and VG, he mapped, like this is the chromosome, the DNA, farther apart, and said, okay, recombination occurs 17% of the time here. So then if I look at the B and how it relates to the CN, recombination occurs 9% of the time. So if I put B and VG here, that 9% means that the CN is either 9 units this way or 9 this way. And so he chose this way. Why? Because he had some data that talked about the distance or the recombination frequency between CN and VG. CN and VG also occurred 9% of the time. So therefore, the CN can't be over here because then that would mean that between CN and VG, that would be much greater than 9. All right, and so therefore it has to be in the center here. And so what this does is tell us relative distance in relationship of these genes to one another. It's not exact. It doesn't tell you exactly where it's on the chromosome, but I know based upon this data that the genes are on this chromosome in this order, and that the Cinnabar gene is basically almost right in the middle of these two genes based upon that. Notice that the math doesn't quite add up here. It says 9% and 9.5 and 17%. And that's okay because the data is not exact, all right, but it gives you relative positions, all right? And so that is a linkage map. So that's what Sturtevant did. He used these recombination frequencies to make these linkage maps for fruit flies and their genes. Now we have more technology. We can sequence DNA. We'll talk more about that later. And so now we can do things like cytogenic maps where we can actually put genes and figure out where they actually are on a chromosome. Linkage maps don't tell you that. Linkage maps just shows you approximate. But we do have technology to make these cytogenic maps to put genes on particular spots of the chromosome. All right, so you know exactly where they're at. And you can put them in relative position to other chromosomal features, like how far away they are from the centromere and certain banding patterns and so on. And then this is just another linkage map, all right, that shows that. So what I'd like you to do is, off to the side, in the margin off to the side, is I'm going to have you do a linkage map I just made up 4 different random genes, gene A, B, C, and D, and I give you recombination frequencies between the genes. And what I'd like you to do is to draw a line that represents the DNA of the chromosome, and I'd like you to give me a linkage map of not only the order of the genes, but relative distance from one another, where they would be on the DNA from this. on this here, you should have gotten, so B and A relatively close together, and D and C relatively close together, because this is 2, I think, apart, these were 3. And then you can do the arrows between the 2, kind of like the example did, but that's kind of the order. Although you could have, like if you picked this up and flipped it around, you could have had C, D, A, B. That's the exact same thing as what I have up here, okay? All right, so if you imagine 3-dimensionally, you can move that DNA around. So that's how you do that. And so it gives you, like I said, relative position. So you know the order, and you know that A and D are farther apart than B and A, based upon the frequencies. But we don't know exactly where it's at on the chromosome. All right, got that? All right. All right. So now let's move on. Okay, so we only have one last section of chapter 15. We already did 15.3, so I'm going to skip ahead to after the calico cat. Got a pretty slow going on outside here. Gotta be positive here. All right. So this last section with chapter 15 is about how we get chromosomal disorders. We've already talked about some ways to get a chromosomal disorder, and the ways that we've talked about so far is inheriting a gene from your parents. So we've talked about recessive disorders, where you get a little r from mom, a little r from dad, your little r, and you have cystic fibrosis, let's say. Or dominant disorders, you get a big R from mom, a little r from dad, your big R, little r, and now you have Huntington's disease. Or a sex-linked trait like colorblindness and things like that. Those are all getting genes from your parents and your parents passing those traits on to you. This section here talks about 2 other ways in which you can get a genetic disorder. One has to do with abnormal numbers of chromosomes. So for humans, we're supposed to have 46 chromosomes. But we're going to see that there are some humans that have 47 chromosomes. There are some that have 45 chromosomes, so they're missing a chromosome or an extra chromosome, and so on. And so depending upon which chromosomes, Roman, that you have, missing or extra, remember every chromosome has different genes on the DNA, and so whatever genes you either have extra of or are missing gives you different symptoms, which then will give you a different genetic disorder. So that's the first thing, is the abnormal number of chromosomes in terms of the number. The second thing is that you might have the right chromosome number, you have 46 chromosomes, but there might be something wrong with the structure of the chromosome. So for instance, there are situations in certain diseases where whole parts of a chromosome are missing. They're deleted out. And so you're missing that DNA. And then that caused whatever DNA that you're missing, whatever genes are on there, you're deficient in, and therefore have certain symptoms as a result of that. So that's what it means by structure. So what happens is this large-scale chromosomal alterations often lead to spontaneous abortions or miscarriages. So I talked about this a little bit when we talked about meiosis. So when we go through meiosis, we're supposed to reduce the chromosome number. So eggs have 23 chromosomes, sperm have 23 chromosomes, baby will have 46. If we didn't reduce the chromosome number, you'd have 46 and 46 in egg and sperm. You'd have a baby with 92 chromosomes. That's what we're talking about. That's chromosomal overload. You'd have 2 of every chromosome from mom, 2 of every chromosome from dad. You'd have 4 of every kind. Mom wouldn't even know she was pregnant, the zygote would die, and that's what that means, large-scale chromosomal alterations. So you can't have like lots of different chromosomes, but we as humans can survive with an extra copy of one chromosome. And not all the chromosomes, but certain kinds you can survive, or missing a chromosome, and so on. And so we can survive with. And so how do we get an abnormal chromosome number? Through a process called non-disjunction. Non-disjunction, the pairs of homologous chromosomes do not separate normally during meiosis. As a result, a gamete can receive 2 of the same type of chromosome, and another gamete receives no copy of the chromosome. Remember, gametes are supposed to get 1 of every kind of chromosome in the egg and the sperm cells. So this picture here shows you how this happens. This is 2 situations in which this can happen. So this is 2 meiosis, 1 here and 1 here. So this picture here is showing you non-disjunction, or the chromosomes not separating properly in meiosis 1. The second picture shows you the sister chromatids not separating properly in meiosis 2. So let's look at this one first. So let's say that this is the organism with just 2 chromosomes. So here's chromosome number 1, here's chromosome number 2. The chromosomes are supposed to line up with their pairs and get separated into opposite cells. Notice here the spindle is not attaching from this side, from the right side, and both of these chromosome number 2s go to this cell. So what happens is this cell has, this is chromosome number 1, and it has 2 chromosome number 2s. This has a chromosome number 1, this goes to the cell, but it gets no chromosome number 2s. So then what happens in meiosis 2 separates the sister chromatids. So these 2 gametes are going to have 1 chromosome number 1 and 2 chromosome number 2s. And this one, when they separate, they're each going to have 1 chromosome number 1, but missing and not having any chromosome number 2s. So that's what happens here. So underneath here, it says N plus 1, N is the haploid number. So it has 1 of every kind of chromosome plus 1. It has an extra chromosome. So in humans, what number would be in these, how many chromosomes would be in these 2 cells in humans? These are gametes.
Yeah.
24. Haploid, so they're supposed to have 23 as an extra chromosome, so therefore 24. All right? This one over here, these 2 are missing a chromosome. So they have 1 of chromosome number 1, but they're missing chromosome number 2 because both of them went over to these cells. So this is N minus 1. So if this was a human cell, these guys would have how many chromosomes? 22. We got this one. All right, got it now. So therefore, what would happen is then if let's say this is the mom and a mistake occurs there, and the dad went through meiosis properly, so he has a chromosome number 1 and a chromosome number 2. Look at what happens when this guy's fertilized. The baby is going to have 2 chromosome number 1s. We're good there, but the baby's going to have 3 chromosome number 2s because mom gave 2. All right, and this is what I was talking about when I introduced remember the calico, male calico cat, and he ended up as XXY. Remember that? So this is what would happen, like mom could give 2 X chromosomes instead of 1, and therefore end up with a baby that's XXY. All right, I'll stop here, and then tomorrow we're gonna wrap up chapter 13.
Is this?
And yes, and then the single sheet that you picked up here today is homework. There's only one problem. The first part, the front page, sets it all up, and the back page is the problem. It's about crossing over and linkage. All right, so we'll redo that.
Reading one notebook for all of my classes. I don't really know what for the class, but I have one notebook for all my classes. There was stuff filled up, and I have one to get my books. Because every year we audit it. Mandy,
That's why I brought it out.
is that your homework? No, I
And then they, yeah, draw it out. Yeah. It doesn't matter how you get it. Okay, so the unlinked. So I gotta go to Target. Target has
I'm not waiting, Romy.
but they don't have.
I got somebody waiting on
buy
me.
one get
Oh,
one
cool. It was his own fault. He kept
I'm glad
like
you pulled those up because I totally
he
forgot.
scratched when he hit the
These are limited
air.
edition. Why is it too? Why are the jeans like full?
They do. I don't know.
can't do this.
Thank you.
I love you this one. And I thought.
There's a black.
Okay. Okay, so how you would work with this? Okay, so we do it out here. So that's this one. So what you would say is this dimension is one of these. See, this chromosome and this chromosome, they're gonna be the same cause. So that parent is great.
Just stay here.
And then that's it.
Just like wing it.
All right, and then there's one. And that's the only thing that you can say. So this is like, all right,
It's just
all right, follow me. Could you use a different color?
Mrs. Walker, do you want me to stop your phone?
Oh, thank you.